All right, let's get this last one. Okay, good morning. So it's really great to see you again. It's been a bit long, a vacation. Hope you enjoy it, or you were preparing for exams. All right, so we had the programming assignment due within the last week. Did all of you submit it already? Okay, that's great. Not yet? Today? Yeah, please do your best to submit it. So that we can move forward with the next programming assignment, all right? And probably we'll have 3 lectures more, and then you'll have some time to prepare for your review paper. So I will give you some time to work on it, okay? Hopefully it will be enough time before the final exam. And the last thing, you'll prepare presentation for your paper, and then you'll present it in the class, and that will be your final exam. Okay. We have 1 to 2 more programming assignment, one of them on the sequence alignment, the other one on pre-alignment filtering, okay? These 3 lectures. It will be easy one, just directly apply whatever algorithm you like, okay? Just implement it. Hopefully you understand how it works based on the lecture, and then implement it on your own, okay? And that will be the end of read mapping. So hopefully I will send you a PDF soon, within this week, that will describe everything you need for the programming assignment. I'm thinking to merge both of them, 2 programming assignments in one of them, and then give you more time to work on it. But it's up to you. If you prefer to have one after another, we could do that as well, okay? We will discuss it over iCollege. All right, so for today, we are jumping from sequence alignment topic to a thing we call filter before you align, okay? So let's do a recall for the things that we were discussing in the early lectures. So our goal in genome analysis is to get a genomic sample and then convert it into the full genome or genomic sequence. And by having that guy, the entire sequence, we are able to compare sequences together, and then we could infer something related to genetic causes for a disease or some phenotypes. Again, it's not necessarily about disease, for example, eye color, hair color, and so on, okay? By having this, we could have a lot of reveals, a lot of information that we could know about ourselves and our living. The problem is that we don't have any mechanism so far that move us from this all the way to that. And we've been trying since 1977. There are a lot of tries in that regard, but we still can generate just fraction of that genome. We cannot give the complete sequence of the DNA. And we've been discussing a lot about sequencing, including short reads, long reads, all the technologies that exist up to now. All of them give you what we call reads, genomic reads, and that based on movie or some electrical signal, some pictures, convert them into reads using a step called base calling that call those bases. So each of those information or data need to be converted into information that is the genomic reads. And now the problem becomes how we can connect all those pieces together to build the puzzle and convert it into the full picture so we can see everything happening in our body. And for that, to solve the puzzle, we need the reference picture, as in any puzzle that you would get commercially. However, if you would like to study the entire population, for example, in this study, they were aiming to study about 50,000 Icelander genomes, then they need about 4 million CPU hours. That's a huge workload to be handled. That's just to compare the genomes together and see what's happening with the 50,000 Icelander. If we want to talk about viruses and bacteria, not a human genome, of course, the genomic workload itself, each genome, is much less in size. We're talking about thousands of characters instead of billion of characters in a human. However, still, if you want to do the genome assembly for about 3,500 genome related to the viral, then we need about 28,000 virtual CPUs. And that's a huge workload. Now, if we talk about the entire world, then good luck doing that. For that, we need to do a lot of optimization in the software. We have been studying through the course a lot about read mapping, but still, there are a lot of space to do optimization and to do the things faster. One of them, we've been discussing a lot in indexing, how we can reduce the workload. We don't want to store all the extracted seed in the hash table because that is growing the size of the hash table, makes it much larger than the genome itself because we're extracting all the overlapping seeds. So imagine, we talk a lot about the number of seeds extracted from here. It's basically n, assuming n is the reference genome size, right? Minus k, and then plus 1. And that's many of the seeds. This is the number of seeds extracted from the genome. And then each seed, let's assume the length is about 15. Then the total length of all extracted seeds will be 15 times n minus k plus 1. Let's say, for simplicity, this is n, right? And this will be 15 times n. So it's much longer than the genome itself. And those are the seeds will be stored in here. So imagine if we talk about much longer seed length, then that will be huge. Okay? So that's one way of optimizing the number of seeds stored in the hash table to make the index or the memory more efficient. But also, there are a lot of data structure to optimize the hash table itself. We could also move from hash table to something else, like Wurst-Wheeler transformation. We didn't discuss it in this course, but hopefully one of you will have a review paper on it. Okay? And now, also in the seeding part, there are a lot of optimization on how we reduce the number of seeds. Okay? And when we jump to sequence alignment, we see several work on how we could reduce the number of calculated cells in the dynamic programming table itself. So we move from the full dynamic programming table into what we call banded, into something else, right? Including the wavefront, e-wavefront, and we talk about the sparse dynamic programming and so on. So if you forgot what are these, so here you can see the reference for the lecture where we discuss several ways on optimizing the dynamic programming table itself. Okay? All right. So now let's go back to our read mapper. Please remember this slide is very important for your programming assignment. Now imagine we have a reference genome. This is given to you. Imagine we have a single genomic read. This is also given to you. And your goal is to identify the location of this, or guess basically. You want to guess it, right? We cannot be certain about where this is coming from because this is really randomly extracted from the reference genome, and we have a duplication along the reference genome. So the exact same read could appear here, could appear there as well. So we make a best guess. So your goal is to guess where's the optimal location of this genomic read from the reference genome. So what we do, we extract those seeds, assuming we are going to extract only 3 seeds out of this read. And then we try to match them with the seeds extracted from the reference genome. And assuming we got a lot of hits or matches. And then our goal is to try to play with those seeds, try to play with this information, and try to calculate what would be the optimal location. And we we decide to go for simplicity, for the simple decision that if we have the exact same order as they appear in the read, then that is probably will be our optimal location that we are looking for. And this is how you should program it. Okay, this is how you should implement it in the programming assignment, assuming you have the blue, red, green. And of course, we are not looking for strictly seeds that are next to each other. It's okay to allow for some gaps between the seeds, right? As long as they don't exceed certain length. For example, if this is entire length of the read is 100, then we could allow for starting from the first match all the way forward as 100 characters, okay? But we allow for those small gaps to just allow or account for some variation. Could be insertion, deletion, substitution, and so on. So we talked already a lot about this. Now, once we identify that location as a good guy, then what we do is basically starting from the blue, assuming this is the first character, move forward with many characters as long as the read length over here, and then have that sequence, okay? So we extract it from the reference genome. And then we have the read itself. That's another sequence. So this is the read. This is what we call reference segment. Okay. So the length over here is 100. The length over here is also 100. But remember, we discussed that we could allow for some margin over here, like let's say 20. This is configurable. So it's up to you. You can have it as a parameter. Why we allow for some margin? In a case we have insertion or deletions, but for simplicity, we can ignore it for now. We just want to make things as simple as possible. We don't want to go into too much detail. 100 and 100, and that's what we call global alignment. Global alignment. Okay. Global alignment means we are doing alignment between two sequences of the same length. But we talk about Smith-Waterman, if you remember, that could work for variable length. So one of them could be 100, the other could be 1,000. And that's Smith-Waterman. But we said we are not going to discuss it in detail. So this one, the global alignment, is the Needleman-Wunsch. Do you remember the name? The Needleman-Wunsch algorithm, right? Which is global alignment. And then you apply the dynamic programming between those two sequences, and then you generate the CGAR, the location of the edits, the number of edits, the type of edits, and so on, right? This is how we learn it so far. Now, what we want to do is to deattach the sequence alignment. We want to disconnect it from the read mapper, okay? So assuming we already did the indexing, extracting all the seed, and then the seeding. We have indexing, we have seeding. Now we have Needleman-Wunsch, but now we want to disconnect, okay? We want to connect here something that we call pre-alignment filter that do something, okay, that does something between the two sequences over here. And then if the result is good, we move forward. If the result is not good, just ignore this step. This way, we could do some saving in the execution time. Now, let's see in detail how we do that. Okay, so the goal of filtering, basically, is to solve the computational problem that sequence alignment is expensive. And remember, sequence alignment is part of the dynamic programming algorithm, right? And we already go over many examples that show that this is really expensive. Okay, because this is n to the power 2, where n is the sequence length. If 100, then 100 by 100. That's the number of cells in the dynamic programming table. Now, if this is million, then million times million, and so on. But there are ways to optimize it using like wavefront algorithm or banded, adaptive banded, and so on, right? But this is another approach to accelerate that problem, which is accelerate any sequence aligner by reducing the need for dynamic programming algorithm. What does it mean? It means we are going to reduce only the need for the dynamic programming. We are not going to play or alter the algorithm itself, okay? We're not going to touch the dynamic programming. We're not going to reduce the number of cells in the dynamic programming table as we did in the output. We're just going to decide, do we need to perform sequence alignment or not? That's it. By doing so, we are reducing the need for this computation. Hopefully, that decision is correct. If we do a wrong decision over here, then what will happen? Imagine that we already identified the location of this read in the genome, and then by some chance, we decide to not perform sequence alignment, and then we lost that location. And then if there is a genetic variation happening at that location, we will not detect it. We just lose it for good, okay? All right, so let's see the key idea behind filtering. So when we detect the location of the read in the reference genome, now we have two genomic sequences, okay? Now those genomic sequences could be any of two types, okay? Could be first a similar string, so those two share some similarity between them, right? Could be 90%, could be 99%, could be 99.999 something percent, or could be totally irrelevant, totally something we don't care. We just share some similarity, like one k-mer is shared between the two sequences. For example, here and here, yeah, they share some similarity, but they are irrelevant, just noise. Just by coincidence, we match them, right? And that is not a good read mapper. Now, if we have a dissimilar string, totally irrelevant to each other, then we need to ignore them because they are just noise, they are just false positive, just pop up to us, but we don't want them, right? However, if they are similar, then this is pretty much interesting, and we want to calculate further the things that we discussed, which is location of edits, the type of edits, and the number of those edits, right? Now, how we define the similarity here? There should be a certain threshold that say this is similar and this is dissimilar, right? Because similarity here is subjective. Should it be 50% of similarity, 40, 60, 70, right? And that's what we call edit distance threshold. So we're going to use the term edit distance threshold. Okay, this is very important, and you should have it for sure in your read mapper. One of the parameters, you should have it as edit distance threshold. Okay. Now, you could use this edit distance threshold for banded sequence alignment, right? So the exact same parameter can be used in sequence alignment and can be used also in filtering. All right. Now, the edit distance threshold, we will use it as a metric to differentiate between similar and dissimilar. If things exceeding the edit distance threshold, let's say this is, for example, small e, and this normally we use it as uppercase E. So this is the thing that actually there between the sequences. So how many differences between those two sequences? For example, E equal 10. But what is my threshold? What is the thing that we are looking for? For example, 5. Okay? If 10 exceeds 5, and it is, then those probably dissimilar sequences. We don't want them. However, if the differences between the two sequences is 4, which is less than or equal the threshold, then do something good, and we will keep them. So this is expensive. Why? Because if you apply dynamic programming, Needleman-Wunsch, for example, or even the most optimized algorithm, for example, Wavefront, you're still wasting the computation because you apply the entire dynamic programming matrix and you calculate a lot of cells, but at the end of the day, you just trash all the computation because they are irrelevant to each other, and I'm not interested in the result. I'm not interested in the location of those variation or the type of those variation. That's why we call it expensive, because we calculate the things that we don't need, and at the end of the day, we just trash them. So why we did the calculation? And this calculation, again, is not simple. It's very expensive. So the idea is to apply some filters in the middle between recognizing the location, the reference genome, and sequence alignment. Okay. However, if we want to apply this filter between the seeding and between the sequence alignment, we should have, or we should observe, three main properties. Those very important properties that should maintain them along the filter. The first one, we want to filter out most of incorrect mappings. What does it mean? Incorrect mappings are the things that exceed the edit distance threshold. If we have any genomic sequences that are different from each other or dissimilar from each other, then we want to filter them out. And our filter should be able to filter out most of them. Now, you could ask me, why most of them? Why not all of them, right? That will be something really optimal, something nice to have, but we don't have it so far. The top optimal algorithm is really sequence alignment. Sequence alignment is the only algorithm that can tell you really quickly what number of variation you have between the two sequences. Okay? So if we are able to detect that number correctly, then we could filter out most of them. However, most of the filters that we are going to visit over the next lectures can approximate that number, but cannot tell you the exact number. Okay? That's why we are aiming to filter out most of incorrect mappings. The second property is to preserve all correct mapping. And here it's very important to note the all of them. What does it mean? It means when we have similar sequences, we should keep them, because those are the things that we are looking for, the things that cause disease, right? So we should keep all of them, but for the wrong one, we should filter most of them, okay? Now the third one, want to do it quickly. Why? You remember why we de-attached the sequence alignment? It was the goal to accelerate this step. This step is very expensive, taking a lot of time, right? We want to not replace it, but having something in advance called pre-alignment filter, right? And this thing should decide quickly, should we go for this or not? Imagine that decision is taking very long. It's taking really long time to decide whether we should go ahead or not. Then we are slowing down the entire pipeline instead of accelerating that pipeline. That's why we want to do it really quickly, much quicker than what we have in sequence alignment. Now, if we maintain or achieve these three properties, then we have a successful filter. Okay? Now let's see an example how those filters work. The first algorithm for today will be Sneaky Snake. Okay? That's published in Bioinformatics in 2020. It was part of my PhD thesis. No, my advisor did. It was Greedy Snake. That's my name. And then the advisor changed it to Sneaky Snake. All right. So the key observation here, if you remember from the sparse dynamic programming, if you still remember what is sparse dynamic programming, where we have, do you need this side? Can I erase it? Okay. Okay, so if you remember sparse, I think I have one slide for sparse dynamic programming, but if you remember, what is it? We have one sequence over here, another sequence over there, and then we identify all the matches, right? For example, there's a match here over the main diagonal, and then some insertion or deletion happening, and then the matches will detour to something else, right? To another diagonal. And then another match over the other diagonal, and so on. So when you look at it, you say, okay, so it looks like whenever we have a variation, then we break down the very long matches into more pieces, right? So if we don't have any variation at all, no variation, then what will happen? We'll have the matches in the main diagonal, as simple as that. Even if we have substitution, we still have the long match on the main diagonal. The moment we start having those variation, then we are cutting down this long matches into smaller matches, right? And now, what is the number of those matches? So every time we have variation, we break it into two. Another variation, two. Another variation, two, right? So based on that observation, we said correct alignment. What does mean correct? Correct means the similar sequence that we are comparing to, and that is below or equal the threshold, right? That is a correct alignment, is a sequence of non-overlapping long matches. So if we are talking about an E equals to, for example, 1, 2, 3. So if we talk about here 3, and then we have 3 variations, then this is correct alignment. And then we notice that it's always about segments that are non-overlapping in continuous movement. So we move from 1 to 2, 3, 4, and so on, whatever the number of the edit distance threshold. And if you look in here, this is genome to genome, very long sequence. Okay, this is alignment between genome and genome, and any dot in this graph represent a match, character to character match. Okay, so regardless if it is blue or red, so this dot, for example, represent a character match from here all the way to there. Okay, and so on. And now if we look at the correct alignment, you'll see the matches are scattered around the main diagonal over here. Of course, you will see some flip over here. What does mean the flip over here? It represents, for example, this sequence and sequence, to represent that this genomic sequence is flipped. Okay, the first reverse, not reverse complement, but reversed. Okay, so the first character over here is last, the last character over here is first. So this one, normally they represent it this way. Okay, when you see this figure, it means this is reversed, not reverse complement, just reversed. Okay, and that's happening by some diseases. Okay, so it's important to observe those matches. Now, this is a very old mechanism published in 1985 called the dot matrix. And you can infer why they call it dot matrix, because each match is just a dot in the matrix. Okay. Now, we already seen this in the anchor-based edit distance, if you remember, from the very last topic in the previous lecture, where we calculate the mums. You remember the mums? The maximal unique matches, right? So this is also the same observation where we have a genomic sequence, another genomic sequence, when we represent all the matches, regardless if this single match or Kamer match or even maximal unique matches, we still have that long matches scattered around the main diagonal. Okay, now this is important for us because also for sequence alignment, we are exploiting that observation. This is sparse dynamic programming, which is the things we talk about here. Now, even if this long match is not as long as the connection of the next match, we still have it scattered around the main diagonal because we are going to apply dynamic programming at that portion only. That's why we call it sparse DP, right? Likewise, we are going to apply dynamic programming over here, dynamic programming over there. Okay? Now, given all those secrets or information that we have in mind, how the correct alignment would look like, we said our goal, or the goal of Sneaky Snake, is to quickly and reliably approximate edit distance between two sequences by calculating only the anchors, which are the things in here. So the observation is just to calculate those, ignore the dynamic programming part, and then make a decision based on the length of those guys. If the length of those guys sufficient to say something about the similarity, the longer matches we have, the higher the similarity between the two sequences. So if we could calculate that much quicker than sequence alignment, we could make that decision very fast. So when I approximate a distance calculation, and based on something called single net routing problem in VLSI chip. I wouldn't be surprised if nobody heard about this problem before. VLSI chip. Okay, that's great. That's why I have this example for you. So this is a chip, right? CMOS chip that exists in every electronic, mobile devices, laptops, computer, and so on, right? Now, if you dissect, first of all, this is the chip itself, okay, the black part, okay? Those are soldering legs. We call them leg, right? Just leg for walking, right? The purpose of this leg is to connect the chip itself with the outside. The outside world could be IOs, like input-output, or it could be another chip connected to that chip, okay? Now, if you dissect through this the black box over here, just remove the outer layer, what you will see basically is this. This is the internal body of the chip. Those are the legs. Okay, it's not interesting for us. What is inside is more component to do some calculation. Those components could be anything, right? Could be some devices that analyze the voice, and then some computation, and then some memory device, and then some more caches and so on, okay? But the thing that we are interested in is how we would connect a leg to any of those devices, and how we would connect those smaller parts into another part. For example, I want to have this as input and any of those as output. For example, I want to have input voice from here using microphone or whatever, and then analyze the voice over one of those components, then send it to another component, do further analysis, and then send it as a sound, okay? Now, how I would do that optimally? There are some constraints that you cannot cross over the components. So if you want to connect, you have to go through the component, connect the input over here, and do some calculation, and then link the output over here to another input, and so on. So you cannot jump from one component to another. You cannot bypass it, okay? Now, it's easy to do it for one connection. I want to connect this to that, okay, it's all clear over here. You could detour the link, whatever you like. Now, the moment you start increasing that number of links, want to connect the next leg, for example. I'm not sure if you can see those green links over here, and the purple, the yellow. Can you see it? Okay, perfect. You can see that if we have a connection over here and want to bypass over that, we want to connect, for example, this component. We don't want to connect this. And then we want to detour over here, and then this portion of the chip starts becoming more crowded, right? And that's more challenging. Sometimes we want to detour from here, want to connect over here, going up, and then we connect this component to that input. Why? Because this entire area is crowded, okay? Now, what we want to consider only is single net routing problem, okay? Single net means want to connect only one leg over here to another leg over there, assuming there are some obstacles. The obstacles here are those components themselves, okay? Each of those components is considered as one obstacle. If it's not interesting, want to avoid it and find another link or another way to connect it, assuming we are bypassing those obstacles. It will be clear in the next slide. And here's the part why we call it a sneaky snake, okay? assuming that instead of we having electrical signal passing from one leg to another, now let's formulate the problem as we have a snake. We want to pass from one point to another point, and we have a lot of obstacles we want to avoid and cross over and find the shortest path from one problem to another. Now, why shortest path? If we are talking about VLSI design, which are the chip we have, every path you decide to visit, that is a cost, extra cost. Why? Because it requires to add a metal layer and then soldering this leg with another component, add a metal layer over here, and then if we want to visit from here to this point, again, add a metal layer, do soldering, and move on, okay? So every step we decide to move is extra cost for us. Of course, this chip is very small, and then the cost is minimal, but it will add up because we are talking about larger scale. So companies don't produce one chip, produce in large quantity, like thousands, millions of those chips, right? And every single cost add up to the chip cost, it will be directly affecting the consumer cost. Now, this is the algorithm itself, okay? We call it sneaky snake. And now let's talk about genomic sequences, regardless of the VLSI chip problem. Given two genomic sequences, which are the thing that we would like to detect their similarity. Are they similar or not? Okay. Now we have a reference sequence, let's denote as R, and a query sequence as a Q. The length go from 1 to M, so they have the exact same length, right? And an edit distance threshold that is E, which is the thing that we use to differentiate between similar and dissimilar sequences. We calculate the entry Z, I, and J, which is similar to dynamic programming matrix, right? So any entry in here, for example, this is I equal to 2, right? Because we start from 0, 0, 1, 2, and then 0, 1, 2. So 2 and 2. If we want to calculate this, then 2 and 3, right? That is Z, I, and J of the chip maze, where I, the thing with the number of rows, varies from 1 to 2e plus 1. So the width or the height of this matrix is 2e plus 1, okay? And that is the first difference between the sequence alignment and what we calculate in sneaky snake. So the height over here is 2e plus 1, but the width of this matrix is n. What is n? Is the length of one of the sequences, or m. Yeah, sorry. Let's use n for the reference genome and m for the genomic sequences, okay? In here, what did we use? M times m, right? But in here, 2e plus 1 times m, okay? And that's the first step to calculate sneaky snake, is to build this chip maze that is mimicking the entire chip in our devices, okay? Assuming this is the chip that we are working on. Now, how we calculate any single entry in this maze is just to follow this equation, okay? Same as we use in edit distance, for example. We had that equation. If the two characters are similar to each other, do this. If they are not similar, do something else. The exact same thing over here. So we have 4 conditions. We fill it with binaries, only 0 or 1. So whatever the equation over here, if it's satisfied, then 0. If not, is 1, okay? So that will be a binary chip maze. Here only will be zeros and 1s. And that's the second difference between the sequence alignment and pre-alignment filtering, okay? The first one was the size of the matrix, much smaller, because NE plus 1 should be always smaller than M. Why? Because the edit distance threshold, there is no way it could be equal to the length of the genomic sequence. Imagine we have the length of the sequence as 100, and then we have the edit distance threshold as 100. It means all the characters are different, so it doesn't make sense. So this should be much smaller than what we have in the link. That's why the size of the table should be much smaller than we have in the dynamic programming table. The second difference, this is calculated using numbers, right? And those numbers sometimes rely on penalty score that we identify. So it could be, for example, 50 for matches, 100 for insertion, and so on, right? So those could be large numbers, and that require more space in the memory. In here, we have only binary, which could make the entire row fit in one SIMD lane, regardless the number of columns over here, because we have binary. So we could compress this entire number in very small size of memory. All right. Now, this equation might look a bit complex, but now I'm going to show you a cartoon that quickly tell you how you could calculate the entire matrix. This one, okay? Now, imagine we have two genomic sequences. Those are the things that we want to decide whether they are similar or not. You pick one character out of the first sequence, okay? And then you pick 2e plus 1 characters from the other sequence. So you pick one character from here, and then 2e plus 1 other characters from the other sequence. Now, can you infer what is e value based on those highlighted characters? Excellent. So 3. Why 3? Because we have 1, 2, 3, 4, 5, 6, 7 characters. That's equal to 2e plus 1. Should have it somewhere here. So that is 7. And then e equals to 3, right? Now which character I should pick? Is it random? Should I pick 2e plus 1 starting from here, for example? No. It should be the corresponding character as the center, corresponding character as the center. For example, this index is 6. Then I should pick the 6th character from the other sequence, and then e other character from the right, and e other character from the left. Why is that? Do you remember what we discussed in Atlanta? When we compare Atlanta word with Atlanta word, we make some variation to the other copy, right? If we make a deletion, what will happen? All the character will be shifted to the right, because there is now vacancy where we do the deletion. And when we insert a character, we shift them to the opposite direction, to the left, right? So to account for those potential or possible insertion and deletion, we should account also for movement to the right or left. So if there's a deleted character in here, all those characters will be shifted by one step. That's why we are scanning the right and left. Does it exist on the right? How about the left? How about there is no variation at all, which is the thing, the corresponding character. So if there is no insertion, no deletion, we expect this character to appear here. But if there are some variation, this character might appear somewhere in here, somewhere in there, based on how many variations. We have 2 insertions, then we should look 2 steps further, right? If we have a deletion, then 2 steps further of deletion to the opposite direction, right? Now, we do a very simple comparison between all those characters. For example, G and G equivalent, right? What we do, we just fill it as 0 over here. G and A, not similar, 1, which is a black box. G and G, 0, so here 0. G and A, black box. G and G, white box. G and T, 1 and 1, okay? So the 1 is a black box. And the 0, nothing, just empty box. What does it mean? It means a match, a mismatch, okay? A match, a mismatch. And now, how does this relate to VLSI design? What does a mismatch mean in the VLSI design? It means it's an obstacle, okay? Things that we cannot avoid, okay? An obstacle in our way. That means we cannot bypass it and jump over it and continue in our way. We have to find alternative and detour to avoid that obstacle, right? And that is the simplest explanation for sneaky snake chip maze, okay? Even if you don't remember what the equation is over here, so you don't need to worry about it. You just, if you implement it this way, it should be perfect for you. Now the index of that column equals to what? The index of that column equals to the index of this character, okay? And the indices of all of those characters is equivalent to the indices of those columns. How many characters do we have in here? We should have that many of columns, and that will be the M size in this chip maze, okay? How about the height? We said it's equivalent to E plus 1, 2E plus 1, right? 2E plus 1, and this is why it's 2E plus 1. Because we go over 2E plus 1 characters from the other sequence, and we try to compare them with that single character over there, all right? Now, why it's faster than sequence alignment? Or hopefully it's faster. We still didn't see the result, we didn't discuss it, but why we expect it to be much faster than sequence alignment? It's something first relate to the size. Yes, the size of the matrix is much smaller, but there is something very fundamental, which is related to the equation that we use to calculate, okay? If you remember in sequence alignment, any given cell, I want to calculate this one, and they are not similar, the corresponding characters over here, what we do, we check those three cells, right? Either we take the minimum of them as an edit distance, or we take the maximum as in sequence alignment, right? But we still rely on three pre-computed cells, right? So we have to calculate them in advance. And this restricts the parallelism. So we cannot calculate all of those rows in one shot directly, right? We should calculate this entire row first and this second row second, right? But in here, there is no way that we will have any dependencies. If we check any cell, regardless, if it is this one and this one, they are totally independent of each other. Even the one next to each other, this and this, there is no dependencies at all, right? Just compare this character with that, compare that with that. Is there anything restricting me from calculating both of them in the same time? No, at all, at all. Just G and G, G and A, G and G, and so on. So you could copy this character many times, right? Fill another array with this G's. They have another array full of G's, and then just do XOR comparison between them. You could use GPU, you could use SIMD instruction, you could use whatever you like, right? And that is the second source of why we have this much faster than sequence alignment. Now, if I ask you to calculate this column, for example, the third column, what you would do? It's a bit tricky because the 2E plus 1 exceeding the range of the sequence, right? For example, I want to calculate the third column over here. You have the solution already. You say you have black, 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 white, black, 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 right? Let's calculate it all together. So when I check the third column, it means we have to take the third character from one sequence and then compare it with 2E plus 1 characters. So the third should be centered to the third over here, right? And then we take E characters from this side, another E character from that side. And the E is equal to 3, right? So let's take 3 characters. I always have a problem with sliding this up, but let's keep it. Let's assume we have it somewhere here, maybe. Let's delete this. Let's calculate all together. So we have the T, and we have this nice shape. We have 1, 2, so we have 3 from here and 3 from there. Okay. So I want to calculate. This is T. What else do we have? G, A, and then G, right? What do we have to the left side? G and G. What do we have here? Nothing. So let's see how we calculate this column. So this would result in a column, right? So we should have a column that also has 7. We have 1 over here, and then we have 3 boxes and 3 boxes, right? Now the first cell is a result of comparison this with that. We don't have anything to compare with, so we should consider it as a mismatch. Doesn't make sense to consider it as a match, right? Because there is nothing over here. And this is a character. So character with nothing should be a mismatch, right? So that is a black box, or 1, basically. And then T and G is just 1. T and G, just 1. T and T is 0, because it's a match, right? Or white box. Then T and G, 1. T, A, and T and G, all 1s, right? And that will be the result of the third column over here. Does it match? Yes. You could try it on your own to calculate the entire thing. It's a very simple algorithm. Any questions so far? All right, so that is the very first step of sneaky snake algorithm, which is building what we call a chip maze. Why we call it a chip? It's related to the VLSI chip, right? Why we call it a maze? We will see now in the second step. Now, the second step is finding the optimal routing path. The optimal routing path. Now, do you remember this chip maze? The entire thing over here? We have it as is in the next slide, over here. So we'll build the entire thing. If you don't believe it, so this is the third column. You have the third column over here, so all black except this cell is white, okay? So we consider this as our map, okay? And we want to travel from one point to another point given some constraints, okay? Now this should be our entrance. This should be our entrance. We cannot use this as entrance, for example. We cannot jump from here to anywhere from here. We should always start from the left side. And we should exit the map or the maze only from that side, okay? So only we can enter in any of those rows, and we can exit in any of those rows, okay? That's the first constraint. The second constraint, we cannot jump over the black boxes. We can only travel through the white boxes. For example, I see here white boxes. We can travel through them horizontally, of course. Whenever we face a black box, what do? We jump to another row. We jump to another row. Why? Because this is a solid component inside the chip. So if we jump over it, it means we will have a metal layer on top of it, and we do soldering over here and there. That's caused to static connection in the electrical signal, and then it means it's connected already. So that's why we should jump to somewhere else and then move ahead in another line. However, every time we move from one line, one row to another row, we are paying a cost. And that cost is one edit. One edit. Let's see it. So this, what we are going to calculate, and remember, we have the E as 3. What does it mean? E equals to 3. We use it first to know how many rows we should calculate, right? 2E plus 1. However, this is also our limit, how many times we could jump over an obstacle. This is our limit on how many times we could jump over an obstacle. So we're going to calculate an approximated distance, but we have a limit on that number as 3. Let's see it. The very first step is to look horizontally how many steps we could move forward. And remember, we cannot move through the black boxes, so we have to move forward through the white boxes only. So how many white boxes do we have in the first row? Nothing. Here, nothing. 0. Here, how many? 1, 2, 3, 4. Here, 1, 0, and 1. What does that mean? It means we could move here further by 4 steps, which is the best way that we could choose, because it can send us as far as possible with no obstacle at all. If we choose this path, can move ahead only 1 step. And remember, what is the goal of Sneaky Snake? Is to calculate, we should have it, keep it. Do you remember the other figure we draw next to this, which is the dynamic programming table with some long matches? Our goal in Sneaky Snake is to calculate those matches quickly. Now, if we go for short segments or short matches, what will happen? Imagine we have the other example over here, where we have some other matches like this. Imagine our algorithm failed to calculate this, but calculates something instead as this, which is, in reality, if you calculate those short paths, if you choose this short path, it means you are choosing this short match over the very long match. What will give you this? This is going to give you a wrong prediction about the matches or the mismatches. Why is that? Because we said every time we have a mismatch or insertion or deletion, we're going to break the long matches into very small matches. Now, how many matches, how many mismatch we have here? We have 1, 2, 3, 4, 5, and 6, right? How many mismatches we have here? 1, 2, 3. So if we calculate it this way, it will give us 6 mismatches. If we calculate this way, it will going to give us 3 mismatches. So it's very important to select the long match over the short match, but that's going to give us the correct calculation or the correct prediction. And indeed, those white consecutive white boxes next to each other represent those long matches. Why is that? Directly go back to the equation. What does mean any of those white boxes? Means a perfect match, right, between a character and a character. And if they are horizontally at the same location, it means the exact same coordinates of one character matching with the exact same coordinate for the other character from the other sequence. It means, we go back quickly here, means in here, to have the 4th cell in the row calculated, means G and A are compared to each other, and A and G are compared to each other. C and A, G and G are compared to each other. Or if we want to follow the exact same example that we are doing, which we start from the very first column, right? It means G and G compared to each other, G and G, T and T, and G and G. What does it mean? It means those entire characters are exactly appearing in the other sequence. That's why we have it as white boxes. And in reality, it is, right? You can see G and G, G and G, and T, G and G are similar to each other. So we could capture those information from the chip maze that those character, the corresponding character over here, 4 characters, are really equivalent to each other from the other sequence over here, right? And then that was our proxy to calculate those long matches using the chip maze. Now go back to here. So we make a greedy choice that whenever we have the longest path to move us forward, we are going to choose that, okay? Now remember, we face an obstacle toward the end of the path. So what we should do? We should stop, penalize ourselves by 1 step, and then choose another row. So once we face an obstacle, penalize ourselves by 1 step, and then repeat everything. Okay? So we penalize ourselves because we face an obstacle. That an obstacle represents a mismatch. Let's go back. Yeah, we said those characters matching each other, right? But the moment we face an obstacle, it directly represents that there is a mismatch. What is the mismatch? C and A. They're not similar to each other. That's why we got an obstacle over here. But we could have some other matches, and that's why we continue. So we penalize ourselves by 1. It means there's a mismatch. So let's account for it. Okay, we account for it. Now let's jump and look for other matches all over the matrix. So we penalize, move ahead, and now we start calculating. Now the obstacle in here, do we need to consider this column? No. Why? Because we penalize ourselves already with 1. If we account for it again, when we count for the next step, then we are going to repeat ourselves, and then we double penalize ourselves. We don't want to do that. So once we arrive to this obstacle, we penalize by 1, jump to the next column, and start counting how many steps ahead we could move. Okay? How many steps ahead? Here, nothing, 0. Here, 4, 1, 2, 3, 4, nothing. 1, 2, 2 steps we could move ahead. Here, 0 and 0, and so on. Okay? This is very important to ignore or skip this column, the entire column, and then keep calculating next. Now, based on those numbers, which way you should choose? Excellent, the 4. That is a greedy choice. We always go for the highest. There is no reason why we go for the highest in here. We could find another highest and so on, but we always go for that as a greedy choice. Why? Because we want to maximize the number of matches. We don't want to get such scenarios in here. We want to get always the maximum one. Now we go with 4. We face an obstacle, but now let's see what does this 4-step path represent. Okay. Now we say that this path represents the very first 4 character matching with very first 4 character, right? But now they are not in the same path. We are having them in another row. What does it mean in terms of characters? It means once we have the mismatch over here, now those characters cannot be compared with the corresponding characters. Here we compare them with the corresponding characters because they match that location. And that location is in the middle. It's in here, right? In the middle of this entire column. It's a row, but it's a column at the end of the day, right? We rotate it and we save it this way. So it's a column. In the middle of that column, over here, represent that entire row over here. That's why when we said the 4 matches over here represent the very first 4 character compared with the 4 character. However, now when we have those matches, first, they are not in this row. It means we cannot compare the character with the corresponding character. We have to compare them with the shifted version. What is it? Can you infer where are those 4 matches? So, yeah. G and G. So G and G, right? Yeah, excellent. G and G, A and A, G and G, and we have A and A over here, right? So we start with A and A, G and G, A and A, and G and G, right? So we don't compare with the corresponding character, but rather with the shifted version of it. And this is clear also from the chip maze, even if I don't know the sequences. If someone give you this chip maze and we decide to choose that path, we directly could infer that the character are compared with a shifted version. Why? Because they don't appear on the centric path. They appear somewhere else, either up or down. If they appear up, it has a meaning. If they appear down, it has another meaning. And that's related to the effect of insertion and deletion. Each of them have totally opposite direction. Okay? So now we have the A and A, G and G, A and A, and G and G. Okay? And that's true in the chip maze. Now we choose this path. We face an obstacle. What we did over here, we penalize ourselves by one step. And then we should skip that column and move ahead. Okay? We repeat ourselves. We go over that column, not in here, but in here. How many white steps? 0, 1, 1, 0, 1, 0, 0. Now we have ties. We have ties. We have paths, several paths, right, with the exact same number of white cells. Which one to pick? In Sneaky Snake, doesn't matter. In Sneaky Snake, doesn't matter. So we could pick, in the algorithm, we pick the very first one. Doesn't matter. What does that mean? Now, 1, 1, 1, what does it mean in terms of character? So we have 1, 1, 1. What does it mean? First of all, this is second last column. Second last mean I am comparing this second last character from here with some characters over here. And we can see it clearly that TNT, TNT, and TNT are equivalent, right? That's why we have the zeros, 3 zeros over that column. So we have T and T, TNT, and TNT. But remember, we are comparing with 2E plus 1. So this is the centric one, or the one in the center, right, in the middle. So we should have 3 more characters over here. We have only one of them. We don't have 2 more. And 2 more are corresponding to those 2. So we put them ourselves as a black box because they are not matching. So those 2, we calculate them as addition, but they don't exist over here, right? So this is the T, this is the G, this is the T, and this is the G, okay? Now, which one to pick as a match? We said we are going to pick the first one, which is this one, TNT, right? Now, what does it mean in the sequence alignment if we find 3 matches? Means you will see something like this. And the red, you'll see something like this. Okay? When you appear toward the end, you'll see several paths that has the same meaning. The one in the middle, you will have like substitution because it's a variation. The one here, you have 1 insertion. The one down, you have 1 deletion. And then which one to pick? It's still an edit. It doesn't matter, substitution, insertion, deletion. It's still an edit. Why we are sure that's an edit? Because it's not linked with the previous one. The previous one is a segment that's separated from those 3, and then we have another segment. It doesn't matter if you pick this or that, it's still you have 1 variation, right? That's why it really doesn't matter which one you pick. However, it is a problem for sequence alignment because we have a penalty. So the penalty for insertion might be different from deletion, might be different from substitution. So in sequence alignment, it is a problem which one to pick, right? And we always maximize the cost. So based on the equation, it's maximum of any of those costs. But in here, we don't care. Our goal is just to calculate the edit distance or the approximated edit distance and compare it with that threshold. Is it exceeding the threshold or less than or equal that threshold? And based on that, we make a decision. All right, so we choose one of them. Let it be the first one. But again, you could make it the last one. It's up to your implementation. Now, we penalize ourselves by 1 over here. It was 2. Now it comes 3. And we avoid or skip that column and we move ahead. However, the next column after we skip that column is the exit. Then we are done. And that will be the 3rd step over here, examining the snake survival. Okay, that's the 3rd step of the algorithm. Now, imagine we didn't reach the exit. What will happen? If there are more columns to go through, what will happen? When we should terminate the algorithm? We could terminate the algorithm if the approximated, the number we calculate over here, if the approximated the distance exceeds the threshold. It means now it's a 3, so it's okay for us. But imagine we hit the 5 edits. Then we are exceeding the edit distance threshold. Why we should continue? Why we should continue calculating we already know that those two sequences that we use to calculate this entire maze are dissimilar to each other. Then we could early terminate the algorithm, assuming that the approximated distance hit already the edit distance threshold. Or the snake arrives at the destination. We already exceed or exits the chip maze, right? And that's another important difference between sequence alignment or edit distance algorithm and sneaky snake. We could do early termination rather than continue calculating everything until we arrive to this cell, right? And then based on this cell only, we could know what is the edit distance value, and then we could stop calculating, but it's already too late because we calculate everything. But in here, you could calculate very small portion. This is the chip maze. Could calculate only up to here and then stop calculating. So it's very minimal in terms of calculation. But in reality, we don't need to calculate the chip maze, okay? So we don't really need to calculate everything in here. What we need to calculate is only this. Why? We really don't need this entire thing. We need only this. Why? Now, the moment we calculate, yes? Excellent. We cannot move forward anyway, horizontally, we cannot move forward if there is an obstacle, right? So once we hit an obstacle, why we should calculate everything, right? Just an obstacle, so we need to skip. Either we skip to the next column, or we find somewhere where it's vacant or allowed for us to move forward. So only when there's white boxes, we could calculate the entire thing until we arrive at an obstacle. Just stop over there, move to the next row, allowed, okay, calculated, okay, obstacle, stop, move ahead, obstacle, stop, white box, obstacle, stop, and move ahead, okay? And then once this is allowed to move forward, we reach the obstacle, we skip one column, and then repeat. Obstacle, stop calculating, move to the next row, white boxes, keep calculating, doesn't matter how far you go forward, keep calculating until you arrive at an obstacle, stop, obstacle, white boxes, and so on. So that is another optimized way to calculate this entire thing and make it very minimal compared to what we calculate in sequence alignment. So all of those optimization, all of those things enabled by Sneaky Snake allow this algorithm to be, until now, the fastest filter that you could imagine. Until today. So it's been proposed in 2020. Until today, it's the fastest algorithm that you could come up with as a filtering between the sequence alignment. Okay? Yes?
You said in terms of like a tie, it doesn't really matter which one you choose. But in sequence alignment, have penalties, so it does matter. I remember an example you showed in one is like if you modify the penalties, prioritize substitution, or if you want to have more insertion. Could you do the same with Sneaky Snake by like prioritizing substitution by like being on the same row in terms of the size?
Excellent, excellent. That's an excellent question. That's really something nice, something I was thinking about when I developed Sneaky Snake. If you go to the source code published in 2020, or maybe 1 year before, because you submit the code to GitHub and then you submit the paper to journal and then wait for the reviews, it takes some time, right? If you go to the source code of Sneaky Snake, you will see something as a comment toward the end of the code. As a comment, I comment it out, okay? But it's still there. And there, I was trying to extend the algorithm. I was trying to optimize something in it. When we have those ties, does it really matter which one you pick? Yes, it matters. In the example your colleague just mentioned, right? Which is a very important point. Should I go for this or that or that? The problem is not with this decision. This decision has nothing to do, okay? you still can go with the decision and nothing is wrong with this number. Still 1, right? However, this decision is going to impact what is next. It's going to impact what is next. How? Because imagine instead of choosing this path, there was another path starting from this point, another path starting from this point all the way to the end. A path starting from this point all the way to the end. Now, since we choose this path, we are going to skip everything in here, although those are part of the solution. Those 3 white boxes, imagine they are white, right? And they are part of the correct solution because this is very long path. Starting from here all the way to here is just white, right? That should be part of the solution because this is maximizing the matches over here. If I choose this path, I'm going to skip this, this, and that, okay? And probably in here because there is a box, so I will start from here calculating the white boxes in here. Now this is resulting in a different number of edit variation. For example, if I choose this all the way to here, and then I start calculating the white boxes from here, then it means the mismatch is in here. But in fact, if I choose this white path, then it's telling me that the mismatch is in here, not in here, right? And that's going to impact where's the location of those variations, right? But also, imagine now this path, the one we discussed over here, is full of white, not in this row, but in fact in this row. What does it mean? It means the character from one sequence are compared with 3 far shifted characters. For example, it will be maybe a bit far, yeah, in here. If those characters are always matching with the last character, because the white path appearing in the last row in here means any character is compared with 3 shifted characters. So this with this with that, this with this, and so on. Okay? Now, if I choose this one, then the shift effect is canceled. I'm comparing with the corresponding character. So now it's no longer single edit because I'm comparing one character with 3 shifted characters. Means there are 3 edit operations happening, right? There are 3 edit operations happening. That's why the match I'm choosing over the white path are with this and that, and then this and that, this and that. It means there is 3 insertion or deletion happening somewhere, okay? However, in Sneaky Snake, we penalize always with 1. We penalize ourselves with only 1 step, not 3, not 4, regardless the number of matches and mismatches. If we choose a path in here, we should penalize with 3. If we choose a path in here, we should penalize with 2. If we choose a path in here, we should penalize ourselves with 1, assuming the previous path chosen was on the middle in here. Now back to your question. Imagine what we discussed last time. If there is no mismatch, then the matches will be around main diagonal. If we have a single mismatch over here, single insertion deletion, then this long match will appear next to the next diagonal. If we have 2 insertion, 2 deletion, then we have it in the next one, in the third one, and so on. How many mismatches we have? We keep shifting, right? And that should be the penalty we should look for, okay? So that's why in the commented code in Sneaky Snake, I try to do something like that. I try to add the penalty, and based on that, I choose which path. Although all of them appear here as 1, but it's not about that number. I should relate which path I should choose based on the previously selected one. For example, if I want to do optimal calculation, I should always choose this one, not this one, not that one. Why? Because it is on the same path. It is on the same path. What does it mean in the same path? It means only substitution happening, right? And single substitution. That's why 1 step. But if it is appearing in here, it means 1 insertion. If it's appearing here, it means 1 insertion happening. That's why it's shifted here, and another insertion happening. That's why it's appearing over here. So if I move from this row to this row, it means there are 2 steps, and that is a 2 variation. But here, 1 substitution guaranteed. Here's 1 deletion happening. And now the penalty side jumping in and try to affect my decision. Based on the penalty, should I go with 1 deletion or 1 substitution or 2 insertions, right? I try to do something like that, but then I end up calculating all possible options to decide which one giving me the minimum, okay? And that end up something similar to sequence alignment. Because I need to go, what will happen if we have 1 variation, then jump here, 2 variation, 3 variation, 4 variation, 5 variation, and then taking the opposite side. What will happen if 1 variation, 2, 3, 4, 5? And I need to do that every time I stop an obstacle. It's still much cheaper than sequence alignment because I don't do that over the long matches. I only do it around the ties over here. When I choose the next path, should I go with this and that and that? So it's still faster, but I couldn't come up with an efficient way of implementing that. But you will see some tries in the code, but I commented out and give up. And then I said, okay, maybe someone else will find it useful and then use it in their own algorithm to build on top of that. Okay. Now, 2 years later, 2 years after Sneaky Snake, you start seeing Wavefront algorithm, right? So Wavefront exploited this information from Sneaky Snake and tried to do something similar with the long matches. So if you remember Wavefront, we go with the long matches. Whenever we stop an obstacle, we start scanning everything, right? And then we choose another long match somewhere, and then we start scanning everywhere. Do you remember Wavefront? Yeah, the EWFA we discussed last time. And then we choose another long match, and then we scan everywhere, and we move ahead. For them, WFA was sequence alignment. So goal, how we move Sneaky Snake to sequence alignment, okay? For us, we failed to calculate that around this point. So both of us, Wavefront and Sneaky Snake, we could calculate those long matches optimally, right? And the problem on how we choose around linking those matches together. And that's the main difference between Sneaky Snake and Wavefront. All right. Now when we look into the evaluation, first of all, this is a bit complex plot to explain, but please pay attention. In here, we have a percentage of how many sequences or how many sequence pair would share similarity and dissimilarity between each other. So this is the rejected mapping percentage. Rejected mapping means how many out of the sequence pair, so this is sequence, this is sequence, both of them, we call them sequence pair, okay? Imagine you have a lot of sequence pairs, this 1, 2, 3, 4, 5, and so on, okay? Imagine out of those 5 sequence pairs that we need to apply sequence alignment to all of them, 80% or 100% of them are dissimilar. It means all of them are similar to each other, okay? So none of them we should calculate sequence alignment. And then the next number is 80%. I mean, 80% of those are dissimilar. So there is 20% chance that is similar sequence. And then we vary that number. We keep tweaking. How about 60%, 40, 20, and so on. And next, this is the speed of pre-alignment filtering compared to sequence alignment. If the filter is 2x faster than the sequence alignment, what will happen? If 4x, 8x, 16x, and so on was to predict in advance